to say it's not only an honor, it's kind of intimidating to give a lecture in this place. So I'm honored to be here, and I hope I can tell you a story that you haven't heard before. So this is not a talk about Bali. Instead, I'm going to be talking about some work we've been doing over the last few years in different islands in Indonesia. And what I'd like to do, I beg your indulgence, I'd like to show a 14-minute video that sets the stage. And the reason for that is anthropologists often work in rather remote parts of the world among obscure peoples doing strange things. So I find that providing a little context in terms of what we're actually about helps when we get into the theoretical lessons that I think can be learned from this study. So let me fire this up. And so we'll begin then. This is a work in progress, a video for the National Science Foundation. It's just the start, but it'll give you some notion of what the background for this research is. So off we go. My name is Steve Lansing. I'm an anthropologist, and I've been studying the cultures of Indonesia since 1971. I began on the island of Bali, investigating the role of water temples. This is the temple Pura Ulun Danau, located in a crater lake in the extinct volcano of Bedugul in central Bali. I've also worked on other islands like Nias, where there's a very different kind of society. The village of Baamataluo, South Nias, is one of the most interesting tribal villages in the world, because this is a tribal society on the verge of transforming itself into a civilization. These people were headhunters, and their organization was tribal, based on kinship. But they were able to organize hundreds, even thousands of people to drag these giant stone blocks up to the top of this hill, several hundred meters up, and execute the sort of fine stonework that you see on the sides. But who were the ancestors of the Indonesian people, and why are their cultures so different from one island to the next? I've been wondering about those questions since my first visit. To understand the flowering of cultures in these islands, we need to begin at the beginning. The last great prehistoric migration of humans into pristine environments was into the Pacific. The first colonists reached Australia and New Guinea about 60,000 years ago. But these were hunter-gatherers who lived off the land. Some of the islands could not be reached until people learned how to build sailing canoes and grow their own food. So there had to be a second wave of colonization. Many anthropologists think that it started in China. By about 6,000 years ago, the knowledge of how to grow rice and build villages reached the island of Taiwan. These people became the ancestors of what we now call the Austronesians. Some Austronesians sailed south to the Philippines, reaching the islands of Indonesia and the coast of New Guinea by 3,500 years ago. 300 years later, they were in Fiji, and Hawaii by around 500 AD. This theory fits the archaeological data, but recent results from genetic studies have raised new questions. A better understanding of the origins of the Indonesian people is important, not only because people want to know where they came from, but because it can help with the treatment of disease. For example, one of the serious diseases in the tropics is a liver infection caused by the hepatitis B virus. There's no cure, but a vaccine can prevent the disease. But which islanders need the vaccine? There are thousands of inhabited islands in Indonesia and limited medical resources. A few years ago, the Indonesian government restored an old colonial hospital as a center for genetic research. They named it for Christian Eichmann, a Dutch doctor who received the Nobel Prize for discovering the first vitamin. The executive director of the institute is Dr. Herawati Sudoyo, a geneticist and medical doctor. Hera and her research team have begun looking into the geographic patterns of disease in the archipelago. They discovered that one of the genetic variants for hepatitis B appears to follow the route of the Austronesian migration into the islands of Indonesia. But much more research needs to be done. In 2005, Hera asked me to join her on a research trip to Sumba, one of the poorest and most remote islands in the archipelago. Whenever they go to the field, Hera and her team coordinate with local public health clinics, but medical resources are limited, and the clinics have to prioritize. We're headed for the village of Momboro on the north coast. We're also thinking about how to explain what we're doing to the local people. As well as hepatitis, we're interested in the local dynamics of other diseases, like diabetes and malaria. Last night, a public health nurse came on his motorcycle to invite the people of the village to gather in one of the clan houses.
Dan itu adalah Profesor Stephen Lansing, orang setengah Bali dia, karena sudah lama sekali ada di Bali, jadi bisa berbicara bahasa Indonesia juga, asal Amerika. Maksud kami ke sini adalah sebenarnya untuk meneliti dan kemudian juga memeriksa beberapa penyebab dari penyakit malaria. Tapi bukan dari malarianya sendiri, tetapi dari kitanya sendiri. Kenapa kok sebagian ada yang sering kena malaria, sebagian lagi tidak. Misalnya saya dari Jawa Timur, beda apanya ya. Orang Jawa itu banyak sekali yang sakitnya misalnya sakit gula. Sakit gulanya sebabnya ini, gitu. Jadi kita cepat tahu kalau saya tanya, Bapak dari mana? Jawa, sakit gula. Oh, kalau gitu saya udah tahu yang mana yang saya harus periksa. Bapak orang apa? Sumba. Sumba daerah mana? Barat, timur. Saya cepat tahu. Jadi ini gunanya untuk menetapkan penyakit dengan cepat. Sebenarnya itu.
Hera wants to make sure that the people understand why we're asking them about their ancestry. So she asks if there are any questions.
Sehingga dari yang lalu dimasukkan yang sekarang, apa yang merupakan tujuan tertentu.
Tapi mungkin juga kadang-kadang di atas itu ada yang menikah antara misalnya orang Wanokaka dengan misalnya orang Sabu, atau Wanokaka dengan Loli. Jadi kita tahu itunya, bahwa hasilnya nanti, oh kita kalau dia agak beda, oh ini rupanya Loli tadinya gitu. Jadi
we're concerned that if it became known that someone has a hereditary disease, they or their family could be stigmatized. So Hera explains that the results of each person's screening exam will be given to them in private by the public health nurses. Everyone needs to understand and decide if they want to give their consent.
Nah, di formulir yang terakhir itu bapak-bapak diminta untuk menyatakan persetujuannya. Jadi bahwa saya memang bersedia diambil darah untuk diperiksa. Dan saya bersedia juga mendapat hasilnya. Gitu. Jadi bisa tanda tangan Bapak atau juga bisa cap jempol. Tidak perlu berhenti.
One of the first results from the Eichmann lab was the discovery that one of the genetic variants of hepatitis B is widespread among the descendants of the Austronesian voyagers. But on Sumba, these descendants are in the minority. They're clumped together in just a few villages. Sumbanese villages have a very distinctive style. In the center of each village there are stone slabs that anthropologists call megaliths, burial places for distinguished ancestors. Surrounding the megaliths are tall thatched houses for the clans. Sumba is located in one of the more remote regions of Indonesia, far to the east. About 1,500 miles to the west, at the other extreme edge of the archipelago, there's another island where the villages look very similar. This is Nias. Could this style of village architecture be a signature of the Austronesian expansion? Or is it just a coincidence? We took DNA samples from 108 men in 2 Nias villages and discovered that all of them, 100%, show the same pattern on their Y chromosome as the Austronesian descendants on Sumba. So all of the men on Nias and a fraction of the men on Sumba share a common Austronesian heritage. The villages and megaliths on Nias look something like the ones on Sumba, only bigger. I'd already done some research in Nias in the 1980s. In fact, it was the similarities between Nias and Sumba that first got me thinking about this project. The upright stones are male symbols, symbols of high chiefs, but the stone benches are seats for the ghosts of the ancestors who come to participate in the great pig feasts. These pictures were taken around 1907. They show giant blocks of stone which were dragged for miles through the forests and pulled up to the hilltop villages where they were carved into monuments for the chiefs and the gods. In Nias, young men have found an unusual use for their megaliths, something that doesn't happen on Sumba. But Nias is atypical. It's very unusual for a whole population of men to be descended from a single male ancestor. We think it means that this island was uninhabited until the Austronesians arrived in their sailing canoes. Where else did the Austronesians go? Flores is the next large island to the north of Sumba. The villages here resemble those in Sumba and Nias, so we decided to take a look. We've reached a village called Bena in Ngada, which is in central Flores, and it may be the most interesting place for our study. The village right behind me looks like a very typical Austronesian old village. It could almost exist in the island of Sumba. So one of the questions that we've got is, are they connected? I mean, how old are these villages? Were they settled by the same people at more or less the same time? What are the connections between these communities way up in the mountains of Flores and on the island of Sumba? This village of Bena in the mountains of central Flores looks a lot like the villages in Sumba. We've got the houses with the tall thatched roofs and the stone tombs to the ancestors just in front of them. But there's an important difference. The villages in Sumba are patrilineal, the men are in charge, whereas here this village is matrilineal. It means that the houses are owned by women, that both men and women inherit their rights to property through their mothers, and so the matriline is stronger here. The question we want to ask is, what difference does it make?
Biasanya, Bapak, kalau yang diturunkan berdasarkan ibu, itu warisan biasanya diberikan lebih banyak kepada anak perempuan, misalnya. Nah, apakah di sini juga terjadi hal seperti itu?
Ya, memang ini sudah turun-temurun sejak dari kami punya orang tua.
Ya.
Tapi yang perana adalah perempuan, sedangkan bagaimana perempuan itu untuk pengeluaran atau omong apa-apa, kita harus satukan dengan saudara-saudara orang tuanya itu.
In Bena, women have a lot of power. It's one of a handful of matrilineal villages where women own things like livestock and land. But these matrilineal villages are quite isolated. They're surrounded by cultures where men inherit power and wealth. We're on our way to one of those villages, a place called So'a, to meet a young doctor.
Nama saya Dr. Paulina Hendrina Helena Peletimu. Saya bekerja sebagai dokter PTT 2 tahun di tempat ini. Sekarang sudah 1 tahun saya bekerja di sini.
The reason we're here is it's a village of people who trace their descent through the patriline, very close to the 2 villages that we've already studied which trace descent matrilineally. So we've been trying to ask some questions to the people here, get some impressions about what difference it makes if you are born as a woman into this village or into the one in which descent is traced through the mother.
Misalnya mama ketemu sama orang Bejawa, perempuan juga orang Bejawa, terus pernah tidak baku bicara tentang hal itu, mengungkapkan perasaan lah, berbagi perasaan tentang hal itu.
Sering kalau antara ibu dengan ibu ketemu, biasanya bertanya, mengapa ya tradisi soa itu berbeda dengan orang Jawa? Itu biasanya bilang, ya itulah perbedaan, karena tradisi terbawa terus, tetapi ada ibu yang berontak, mengatakan, tidak, kita mesti galangkan itu. Tidak boleh kita wariskan kepada anak-anak itu demikian. Jadi wanita dan laki-laki itu sederajat. Biasanya.
Iya, maksudnya sekarang anaknya mama semuanya cari jodoh sendiri, tidak dijodohkan kayak mama.
Mama itu anak kita, satu lain, satu anak lain, tapi dicari sendiri.
Waktu masih muda, mama dijodohkan. Tapi sekarang dia punya anak-anak, semua sudah cari jodoh sendiri. Maksudnya tidak dijodohkan, mereka cari calon sendiri. Terus kalau mereka sudah dapat calon, mereka kasih tahu mama supaya mama masuk minta. Ya, masih
Pak. Belis di sini jodohnya pakai syarat. Ada soa budaya soa. Di sini ada syarat anaknya mencari jodoh tapi punya syarat.
Masalah belis, kalau sudah suka sama suka, terus lapor sama orang tua, terus yang tadi dijelaskan sama bapak, bilang kalau dari keluarga laki-laki nanti bawa kuda, kambing, ayam, kerbau, terus nanti dibalas dari pihak perempuan bawa beras, kain, babi.
Change is coming to some of the islands, but customs like patrilineal kinship have deep roots. But why are things so different from one island and one region to the next? Until now, anthropologists have mostly been content simply to describe these differences. But in the last 10 years or so, we've been given some new tools that allow us to look much more closely into the past. So we're looking at the relationship between genetics and historical linguistics, which sounds complicated, but actually it's pretty simple. On the genetics side, we're looking at little things called microsatellites. So in every little region of DNA that exists, there are things called microsatellites. These are bits of genetic code that appear to have no functional significance. So they're free to mutate, and mutate they do. The number of microsatellites that differ between one person and another is an index of how closely related they are. So the microsatellites are like a little molecular clock that tells us how far back a given pair of people share a common ancestor. And we can push that all the way back for thousands of years if we need to. So on the genetic side, we can use these microsatellites to gauge the relatedness of people within a village or between villages, in one island or between islands. Before we started looking at microsatellites, the story of origins looked pretty simple. The Austronesians sailed down and colonized the islands. But when we looked at the Y chromosomes of Sumbanese men, we found that only a small fraction come from Taiwan. The other lineages are much older. So it looks like small numbers of Austronesian colonists landed their canoes on an island that was already populated. The Austronesians could have spread out from a single village somewhere on the north coast. So we're in the village of Wunga in northeastern Sumba, near the beach, and the Sumbanese believe that this is the place of origin of all the clans. Most of these houses are empty most of the year, but every year in the seventh month of the Sumbanese calendar, people come back to worship their ancestors at these ancestral houses. So their myth of origin comes pretty close to what we think is going on from the standpoint of genetics. That is, there was an initial migration here, and then after that, clans would have formed and moved out across the landscape, settling in the most desirable areas. So the real question that we're trying to investigate here is the literal truth behind the legends of Sumbanese origins. Here's Wunga on the north coast, the driest piece of land in all of Indonesia. The Austronesian colonists brought their genes and their languages with them. When we started this research, it was thought that the Austronesians moved out in a great wave, replacing or blending with the people who were there before. But our new data tell a different story. Austronesian genes are found in small clumps, and the same is true of their languages. Today, some villages have retained a lot of Austronesian words in their local dialect, but others have not. So here's what we think might have happened. Sumba was already populated by hunter-gatherers. They would have been concentrated in the west, where there's more rainfall. The Austronesians arrive, build their village, and as their population grows, begin to create new villages. The villages in the west would have been surrounded by indigenous people speaking different languages. As the two groups intermarried, the proportion of Austronesian words and genes would gradually fall off. But in the east, the Austronesian villages would have been more isolated and retained more of their original genes and language. So the film ends there, although we think that actually turns out to be right. So we've got a paper that's going to be in the next issue of PNAS, essentially repeating the last bit of the film, telling you that story. So let me explain what this has got to do with the neutral theory. We're looking at variation on the scale of communities, and we're interested in where they came from and then how both matrilines and patrilines move around the landscape, because that helps us understand kinship patterns and their effect on the structure of relatedness within communities, which is also related to how language moves and also disease. So we're convincing ourselves that that's sort of the right scale to look at these processes, these interactive processes, but we realize that the tools of human behavioral ecology don't tell us very much about things at that scale. If there are any human behavioral ecologists amongst you, I mean, the idea there is that the dominant theory has been looking at the fitness advantages of particular behaviors. Right? So it is a kind of pan-selectionist approach, assuming that variation in behavior or in pretty much anything occurs as a result of selection, fitness advantages. We're looking at a different scale, and we noticed that that approach has been criticized by prominent geneticists for decades. Dick Lewontin and Marcus Feldman and others have been telling us that you can't assume that selection is the only force that's going on. And indeed, when we look at these little communities, we think that drift and stochastic effects are more important than just selection. So we sort of rethought the toolbox, and that's what I'm going to be talking about next. What kinds of tools are appropriate to understand variation in all of these things, in kinship and languages, basically behavior. So let me launch into that. I'll talk first about replicators. R.A. Fisher's fundamental theorem of natural selection he came up with in 1930, and there it is. I hope everybody can read it. It says that the rate of increase in the fitness of any organism is equal to its genetic variance at the time. That's now known as the replicator equation, and it's become very important to evolutionary game theory. Interestingly, if you ask a geneticist what's the fundamental theorem of natural selection, I predict he may not remember. So it's not so important in genetics, but it's become fundamental to evolutionary game theory, human behavioral ecology. So Martin Nowak, for example, just did his very interesting book based on evolutionary dynamics. It's all about replicators, and it's very little about drift or non-selective processes. They've got a new center for studying evolutionary dynamics at Harvard that Martin has created, and it's about really the triumph of the replicator equation. So I'm going to say a little bit about the replicator equation because his mentor, Carl Sigman, has, the earlier work shows that equation is at the root of Lotka-Volterra. It's basically, it's regarded by evolutionary game theorists as the fundamental equation for thinking about how the world works. So all it means is selection. So here we have these are slides that I've taken from Martin's inaugural lecture when he created that center at Harvard. So we have variation in fitness between A's and B's, and B, with a little higher fitness, will eventually outcompete A. That's what it says. And let me skip on. So the replicator dynamics tell us that the change in the frequency of any phenotype or behavior or individual depends upon the difference between its fitness and the average fitness in the population. And what that'll do is to move the fittest individual to become ultimately dominant in the population. It just outcompetes the rest of them, right? So it's a very simple idea. In the class that brought me to your university, we talked a little bit about that yesterday. So here's a little simple model written in Stella to show how that can happen. We just have I need my little pointer. I was given a pointer and I've misplaced it. Well, anyway, the idea is simply that we have blues and reds, and they're in a population and they compete with one another. The fitter ones, the blue ones, will ultimately go to fixation, meaning they will become the only thing left in the population. And as they do so, the mean fitness of the population will rise. So that, in brief, is the replicator equation. It's a very simple idea, but it's been questioned. As a matter of fact, Sewell Wright raised an objection a few months after the publication of Fisher's original book, 1931, which he said, yes, it's true that the rate of increase in the fitness is equal to its genetic variance. You have to have some variation so that there are some fitter ones that can become dominant. But it can also be affected by mutation, migration, change of environment, and the effects of random sampling. So there's other stuff going on, which in a way takes the mickey out of the replicator equation because it says that there may be many other confounding effects. And indeed, in his recent work, this is an article in a review of evolutionary game theory in Science a couple of years ago, Martin and Carl Sigman observed that replicator dynamics don't actually fit well in the framework of population genetics and predicted that finite population size effects will lead to surprising outcomes and might question the importance of traditional evolutionary stability. If true, that's pretty devastating. Right? It means that we're going to have to rethink what's going on. You can't rely solely on the replicator equation. So the question comes up, are real evolutionary dynamics at the scale that we're interested in, we're now talking about human societies, governed only by fitness differences? The Red Queen effect says that, as the Red Queen told Alice, sometimes it takes all the running that you can do to stay in the same place. So in evolutionary biology, that means that it's possible to have a condition of fluctuating dominance in which first one gets ahead but then the other, kind of arms race. And if that continues, then what you get is not the dominance of a single fittest type, but rather the preservation of diversity. So it raises the question: how much, how much diversity is consistent with some selection happening in a population? And that shifts our attention from the reasons for the fitness of the fittest individual to the consequences of the whole business, the whole competitive system for a population. What's going on at the population level? So that's the question we began to ask in looking at our Indonesian samples. So what I'll talk about today quickly is, first of all, why the geneticists have forgotten the replicator equation. And I'll just point to the critiques of pan-selection, which are regularly made by guys like Lewontin. Then I'll talk a little about the neutral theory, and then finally show you what we actually saw in the communities that we've been looking at in eastern Indonesia. So the story, briefly, and by the way, these PowerPoints have too many words on them. The reason is they're also designed to be used by the class that's been constructed around this. This talk that I'm giving you is the third talk I'm giving in this series for a class, so I wanted to make this available to the students. So pardon me for having more words than you're supposed to have in a PowerPoint presentation. Anyway, so the story goes, 1940s, the geneticists were pan-selectionists. In other words, they thought that genetic variation was due to selection. In the 1970s, John Maynard Smith developed evolutionary game theory, which basically applies Fisher's model of selection to games and behaviors, and it's taken off since then in its own independent trajectory of game theory. But meanwhile, in genetics, Moto Kimura came up with the neutral theory of molecular evolution. He looked at Lewontin's results, which were just then becoming available, and realized that a good deal of this selection, a good deal of the variance that you see cannot be due to selection. And so he proposed that in some cases, genetic variation will be due to processes of drift rather than positive selection. And by the 90s, we knew that Kimura is right. That is in fact how it works in the biological realm. Drift is very important in selection. It's rather hard to find evidence of selection in genetics. In around 2000, Steve Hubble took exactly the same mathematical theory, the prediction with respect to the rates at which stochastic effects will create variation, drift, and applied it to ecology. And I'm going to do my best to explain what he meant, because it's the ecological version of the neutral theory that I think is most relevant to us. So what is the neutral theory? Well, the neutral theory says that most mutations are neutral. That fitness is irrelevant. Moreover, that they occur at a constant rate. So they're large stretches of the genome, like those microsatellites, which are not under positive selection. So when they ping, when a G turns into an A, when there's a little change, it doesn't have a selective effect. Therefore, the rate at which it happens is pretty close to constant, especially if you look at enough sites. That's why we have molecular clocks. Molecular clock works if you can assume that selection isn't happening and variation is probabilistic. So most mutations are neutral, point one. Secondly, that frequencies then are governed by drift, and that means that it's only when you see a departure from the normal, regular, probabilistic pattern that you can infer selection. And that's how it works in genetics. We see you infer selection only by showing that there's a departure from a stochastic equilibrium. And in genetics, we find that selection it's hard to find, actually. They're looking around trying to find evidence for selection. So if that's true, then this is wrong because it's incomplete. This is back to Martin's talk. It's true that evolution needs populations of reproducing individuals. It's true that evolutionary change occurs by mutation and selection. But it's also true that drift and sheer chance also produce evolutionary change. And maybe we need to think about that. So the neutral theory moved into ecology when Hubble applied the same test, which is the Ewan sampling formula, which simply predicts drift is governed simply by population size. So depending upon the size, you get a prediction for the frequency of rare and abundant species in an ecosystem. So the contrast is the selectionist view, dominant until Hubble, is that the frequency of species in an ecosystem is due to their selective advantage. You study a particular organism and you say, what's its function in this ecosystem and how well does it fit in? What are the selective advantages that make it possible to be rare or dominant in a particular case? The neutral view says They're all the same. Assume that they all have, you don't really believe this, but you say, let's assume, what if it were the case that all competitors are equally competent? What would the world look like if you had no selection? Drain the selection out of the picture and look at a world that's governed entirely by drift. So here's how it works. If species often move between communities, then you get a picture that looks like that, in which on the left axis, and this is the kind of the key graph for this story, okay? So on the left, we've got the number of species, and on the x-axis, the number of individuals. So what that picture would show you is that within a particular ecological community, you have lots of rare ones, with a frequency of 1, you see on the left, rare individuals. There'd be lots of different rare types, and then a few that are extremely common. That would be true if species move around and the effective population size is large, because the metapopulation includes lots of subpopulations. In other words, a big population, they can move around. If, on the other hand, they seldom move between communities, you get this kind of picture. Because they exist in small communities, random chance will weed out those rare individuals, and so it'll depress the curve on the left here. You'll have only a few rare species and many more common ones. So those would be the two contrasting signatures for community structure. Here's then Hubble's most famous plot, really, looking at the abundance of tree species in a Costa Rican forest. But just to be clear, so on the left there, what he finds are 77 singletons. By a singleton, I mean one example of a particular species. So you've got 77 unique species on the far left, and then on the far right, there's one species that has 500 trees. Okay? I harp on this because it took me a while before I could figure out this plot, so I want to make it really clear. All right. So anyway, then what you get, the Preston plot, that red line, is the distribution that would be predicted by the UN sampling formula, which I will spare you, but essentially it predicts that kind of distribution, which fits pretty well to the Costa Rican forest data. Okay, so that in brief is the neutral theory in ecology, again assuming what's the world look like without any kind of selection. Here in the social sciences, pan-selectionism still reigns. So the pan-selectionist view, the idea that all variation is driven by selection, dropped out of genetics. It's now being debated in ecology. Hasn't really begun to be debated in the social sciences, but I intend to debate it. So here's Bruce Winterhalter and Eric Smith in their textbook on evolutionary ecology, and they said, just to define how do you do evolutionary ecology, that an analysis takes the form of asking the following question. In what environmental circumstances are the costs and benefits of a behavior such that selection would favor its evolution? And that's what behavioral ecology does, right? We try to figure out hundreds, thousands of articles trying to figure out what are the selective advantages or disadvantages of particular behaviors. Another example here is Bobby Lowe. What are the fitness effects of different strategies in particular environments? That's the canonical question, right, for an evolutionary game theoretical or behavioral ecology analyses. And the example that I'm going to talk about is male dominance. Shall I read it? I mean, in more than 100 well-studied societies, clear formal reproductive rewards for men are associated with status. High-ranking men have the right to more wives. So male dominance is ubiquitous, according to this analysis, because the prize for winning the fitness race, if you're a man, is to have more wives and more children. Now, if that were the case, and I should say in the evidence for that, it's not that there isn't excellent evidence for that. There's lots of demographic evidence. In fact, Bobby did a lot of work here in Sweden looking at the demography of historic, I think, graveyards in Sweden, and what she shows is the higher-ranking men do, in fact, have more offspring. Okay, so with demography, you can only get a very short time window to look at that phenomenon, a generation or two. And what I'm going to suggest is if we can now shift the focus and ask, what about the population-level consequences of selection? So I move now to what we actually saw in Bali. I say Bali automatically. I mean Indonesia, the other islands, Bali, but also some other islands. What do we see in terms of evidence for male dominance, selection effect in real communities? Can we apply the neutral theory? the ecological version. So what this means is it's a very simple idea. We're just taking the question of what are the abundance of species in a community and asking instead, what is the abundance of patrilineal descent groups in a human society, right? And you can imagine if it were heavily skewed, so only very dominant men had all the women, then you'd only find one descent line in a village, right? If Genghis Khan marries all the women and all of his children take over the village, from then on, the village would be dominated by a single patriline, namely that of Genghis Khan. So we can imagine that that could be the case. So what we can do is to count the frequency of different sized patrilineal descent groups in a village using those microsatellites. So here what I'm doing is to count the number of patrilines and then the number of individuals who belong to each patriline, each patrilineal descent group. That makes sense? Right? Traced through the Y chromosome. Substituting these patrilines for species. So we looked around Indonesia, as you saw. Little red dots show some of the islands we worked on. It's quite we managed to span a large variety of different kind of cultural types. Up there in Borneo, we have some hunters and gatherers, wet rice farmers in Java and Bali, tribal people in Flores, and some matrilineal and some patrilineal groups. So nice wide range in which we can ask that question. So the way you actually trace the patrilines is just using this genetic data as a marker, nothing more complicated than that. So if I were to take a cheek swab from you and identify we would look at your Y chromosome if you're a male and identify the SNP, the single nucleotide polymorphism, which defines the branch that tells you where your father's ancestors came from. So that's what that looks like. So there's the SNP that defines my branch. I mean, if you send away, at least in the States, it's now becoming popular to, you get a little kit in the mail and you swab your own cheek, mail this thing back to the National Geographic Society. They charge you a few $100s and they'll tell you where your SNP came from. Where did your ancestors come from? So that's what this is. So I belong to that SNP. But we're looking now, we're interested in a finer scale resolution, not just the SNP that defined a major branch thousands of years ago, but rather the little tiny branches that define my relationship to my closest cousins. So using these very quickly mutating microsatellites, you can trace resemblances between people that are only a few generations. It doesn't take very long to see some small changes in these fast mutating regions of microsatellites. If you look at a large enough stretch, you could probably tell me from my son. There'll be one or two little differences that will emerge even within a person's lifetime. Okay, so you get this kind of picture. Everybody here belongs to one patrilineal descent group. Those are what I'm going to call patrilines. So here's a plot then for the village of Kodi in western Sumba. We sampled 43 guys and found 17 haplotypes. That means a patriline, 17 lines of descent. I really do need I'm just going to walk over here. So here's one patrilineal group which is represented by 23 men. So this is a large group of men who are all descended from the same close ancestor. Over here at this end, we've got unique haplotypes. The colors mean the haplogroup. I'm sorry for the confusion of the terminology. What it means is they belong to different major SNP branches. But what we're interested in here at the moment is just how many unique patrilineal groups are there. That's all these so-called singletons. It's as if they were rare trees, unique trees in the forest. These are unique. This is one man, one descent line. And here's one that has four men share the same haplogroup here, and here we've got one very dominant one. So that looks like male dominance, right? I mean, here we have, and in fact, this village does show male dominance, as I'll show you in a minute. But they don't all. So that graph makes sense. Right, so we've got one haplotype shared by 23 men, one patriline, and then these 13 different unique ones. So if the dominant males were really super dominant and didn't allow other men into the village and married the daughters of men in the village at sufficient frequency, the guys on the far left would vanish. They wouldn't be in the village. These guys don't belong to my group. But as you can see in that village, quite a few of them, there are 13 different groups which do exist in that village. So we see a kind of a mixed picture in this particular village. One dominant group and then a lot of variation from people who either migrated in or were were there, but only in small numbers to begin with. Selection. If there were a selection effect, if male dominance worked and the high-ranking men had more wives, that would reduce the variation within the village, right? Leading to the dominance, in the extreme case, of a single patriline. So we can compare the distribution of haplotypes in the village to expectations under either neutrality or selection and just see what they look like. Is there maybe a red queen effect in which one group gets dominant but only for a generation or two, and then another patriline might acquire dominance? We could make up stories about that. I mean, maybe some groups acquire more wealth and acquire more wives, but are they able to pass that on to their grandchildren or their great-grandchildren? So it's an open question. So here then are the three possibilities. If it's purely neutral, the little colored dots are supposed to be men. So if everybody's probability of having children is equal, then you get a picture like that, and there is no change in frequency of the haplotypes except from drift from one generation to the next. If it's a red queen system in which the patrilines compete but they don't get ahead for very long, first one then the next, then you get the kind of picture you see there. You can actually see that genetically because it shortens the time to the most recent common ancestor for reasons we won't talk about now, but you can actually see the signature of a red queen effect. Here's extreme cultural selection in which the dominant male reproduces more, and within 5 generations, it's only his kids. Okay, so the signatures then on the bottom, no change or a shorter time to the most recent common ancestor for a red queen, or drastically reduced diversity under conditions of male dominance. Okay, so what do we actually see? Well, these are simulations, which I hope you can make out, at a neutral equilibrium, just running computer simulations, and you see, right, typically we see quite a few single individuals on the left, and then in some cases, the first and second, you see a kind of a couple haplogroups that are larger, although the two on the far right show no dominant groups. Here, 4 real Indonesian villages, which are all at neutral equilibrium, no evidence for selection. And then here are 4 villages that are not selectively neutral. But these non-neutral villages that you see on the bottom, if you can see them, so here we have dominant lineage. Here's a less dominant lineage. Here's a dominant lineage. I think there's one in there. But these two are selectively not neutral, not because of a dominant lineage, but because they're mostly in migrants. There's too much diversity, not too little. These are very diverse villages. So we can look at both those effects. And what we find is, I think it's the next slide, the punchline. Well, most villages, in fact, 37 out of 41, 37 villages out of 41 were neutral. We're selectively neutral using this test, the Ewan's test. So that surprised us a little because I've been raised to believe in the behavioral ecology story, and yet we don't actually see it here in these villages. So we were surprised, and we began to try to figure out what would it take, how good is this test? Now we have to look at the mathematics a little bit and say, how good is the Ewan sampling formula applied to this question? Because we have to think about time, in other words. So let's assume that the patrilineal kin are competing, and how fast will selection reduce the diversity? So we start here, and within a couple thousand years, this starts with complete diversity in which every male is different, and within a few thousand years you reach a kind of an equilibrium. The reason you reach an equilibrium, I think this is shown better in the next slide, is as time goes on, new haplotypes appear because of mutation. It's not frozen, so you get new variation within a village either by in-migrants from someplace else or just by mutations which create new haplotypes, which happen at a regular frequency. So let's see. Basic idea is that if persons, and you can apply this also to language in principle, if persons or words or genes or whatever move between communities frequently, then you get lots of rare ones. If, on the other hand, you see barriers and the communities are very isolated and small, then the rare ones get weeded out. And so you get much more skew towards a few common ones. Okay, so here's a simulation. This begins, let me see, let me back this up. we're going to start with a simulated environment in which everybody in a village that includes 200 men shares the same haplotype, and then run time forward, allowing mutations to happen at the observed rates of these microsatellite mutations, and see what happens. So what happens is, here we start with, this is, I'm sorry, in this case it's a single mutation. Everybody starts out with the same haplotype, but then as time goes forward, new ones appear, and so quite quickly you reach this point. And at that point you're at a neutral equilibrium in which the appearance of new haplotypes is balanced by the disappearance of other ones due to drift, due to pure chance, and it stabilizes. That's this whole population, and that's the sample. You can also run the same simulation starting with 200 different haplotypes. So this is a village in which every man belongs to a different patriline, but as you can see, quite quickly, drift removes those haplotypes, and you wind up at the same stochastic equilibrium. And it doesn't take so long. It's about 20 generations. Well, depends on how you think about time, but pretty quickly. So this then summarizes that. It's a pretty rapid approach to stochastic equilibrium. Whether you start with all haplotypes different or all the same, you'll get to this equilibrium state, which is what we find 85% of our villages maintaining. Now, 20 generations actually is, in human years, that's actually a long time, right? So it means that if these villages had a dominant male some generations ago, we would still see the effects of that. It takes some time for the effects of selection to wash out. So this is actually a pretty strong case for neutrality being the common, being very common. We're also encouraged that we do see these other cases in which we do pick up the signal of dominance. So in 4 out of the 8 villages that we had then sampled on the island of Sumba, we do see male dominance. Now, Sumba is a society in which men wear swords, frequently go with each other, even now, very strongly patrilineal. They're exactly the kind of place where you would expect to see male dominance. Dominant men, in fact, do have more wives in Sumba. So in 4 out of the 8 villages that we studied, that's in fact what we see. But the real question is, what about the other 4? How strange that we don't see male dominance in the other 4 villages in Sumba, looking at their population structure. So it's also important to note that the non-neutral cases aren't necessarily the effect of male dominance. They could be the result of population bottlenecks or founder effects. So the case for ubiquitous male dominance is looking pretty flimsy, at least looking at these Indonesian villages. Now, it's worth, one more thing to say about this. No, I'll just go on. Okay, so if you think then, but let's scale up and just ask a question based now only on the Indonesian data, because that's all we've got at the moment. What is consistent with the actual levels of genetic diversity that we see in human communities today? If it were the case that male dominance was common and males had a selective advantage, the dominant males had a selective advantage of about 4%, then we would not see the diversity that we see in these communities today. Human beings have been living in small groups really for most of our history, right? Up until a couple hundred years ago. And so the effective population size is quite small, right? A few hundred probably. So if it were in each of these little communities then, if there were a strong bias for male dominance, it would be weeding out genetic diversity. In the Indonesian cases here, this is the time to the most recent common ancestor for the various lineages, varying, for example, the K go back between 13 and 88,000 years. And they're represented only in fairly small numbers in Indonesia. If there were strong male dominance, we wouldn't see these guys anymore. They would have been weeded out of those villages long since. So the sheer diversity that we see in these communities suggests that there hasn't been a lot of selection. And it's also relevant to say that if we don't see selection for male dominance, it means that nothing has been selected on the Y chromosome. In other words, the candidate explanation that we get from human behavioral ecology is rewards for high-ranking men. That's the one we've tested. But in fact, we don't see evidence that we could imagine other kinds of selective events that would also be skewing the distributions in these villages, and that would make them non-neutral, and we don't see that. Is that clear? In other words, male dominance is only one possible explanation for reproductive skew. And since we don't see reproductive skew, we don't need to waste our time thinking of more possible explanations for it because we don't see it at all, right? Now, what I'm doing is trying to tilt then the argument in the direction that it's gone in genetics to say, if you want to make an argument for selection, you first have to show that it exists, that there has been selection, and then having done that, you can start making up stories and trying to figure out what is the cause for selection in this particular case. That's very different from saying, okay, we've got a behavior, what might its possible selective advantages be, right? It just shifts the burden of the argument completely to first demonstrating that there is a selection effect. So here's the conclusion then. Wright was right. The replicator model, the pan-selection model, everything being under selection is too simple because drift and stochastic effects matter, at least based on our Indonesian cases. Thanks to Kimura, we know that that's true in genetics. And thanks to Hubble, we suspect that it's true in ecological communities. You now have to make a case to say there really was a selective advantage for particular species abundance in a particular case. What that means is that the best null model is not to assume selection is constant, pan-selection, but rather neutrality. Start with the assumption of things being selectively neutral, and then look for evidence of selection at the population level. So this first test of the neutral model in anthropology showed that male dominance rarely yields long-term evolutionary benefits. I mean, from time to time, selection must have happened, but it isn't ubiquitous. That's what this shows. It seldom persists in our cases for more than 2 generations, or we would see it in the population structure. And if it did persist, then human genetic variation on the Y chromosome would be much smaller than it is. The next question, of course, is let's look at macrolines at mitochondrial DNA. We started to do that to look what kind of dynamics do you see with respect to descent groups through the mother, right, through the macroline. We hope to find out at least with respect to our Indonesian samples. And then finally, I kind of like this conclusion because it's consistent with my view about. I mean, I have to admit this. I'm happy that what this view suggests is that diversity actually is frequent because the replicator equation produces winners, weeds out the others. But in real human communities, it looks as though the red queen has been busy. Competition doesn't lead to the dominance of a single winner every time. So this work has been, I'm the mouthpiece for a large number of people who've been working on this for several years, Indonesians at the Eichmann Institute, and then the people whose names, I won't read them for you, but the people I mentioned here. And that's my story. So thank you very much.